Hi, let's begin. Our topic this evening is biomarkers, blood tests, and breakthroughs, the importance of early testing and diagnosis for dementia. My name is Tony Teano. I will be your host for this webinar. I work with the Johns Hopkins Alzheimer's Disease Research Center. I would love to know how you found out about the webinar tonight. So my dear colleague Corinne Pettigrew is going to launch a poll, and it will ask you about how you found out about this seminar and through which organizations you found out about the seminar. So you please do take a moment to let us know how you found out about it. This is a very highly attended webinar, and we're dying to know what the best communication tools with you would be. So thank you very much for taking that. And let's see, it looks like I need to check something to get this to go away. Host and panelists can't vote. All right, so I can't vote. I should just close that out. Is everyone else voting, Corinne?
Yep, responses are still coming in. I'm going to give it another moment.
Excellent, that's great. So while that's happening, let me tell you a little bit more about our topic tonight. Again, it's biomarkers, blood tests, and breakthroughs, the importance of early testing and diagnosis for dementia. And you've already heard from one of my co-colleagues here. The moderator is going to be Miss JoAnn Scipio. She's with the Caucus of African American Leaders of Anne Arundel County and also the Annapolis Alumni chapter of Delta Sigma Theta Sorority, Inc. She's also an NCLEX instructor with Kaplan Test Prep and Admissions. And then you just heard from Dr. Corinne Pettigrew. She's taking care of the behind the scenes technology for the Q&A, and she'll also monitor the chat box for you as well as the Q&A session. So I'm really very happy to have them with me tonight. And let's see what else I've got for you. Here's a quick overview of our program. We will have two very distinguished speakers for us tonight. One is with the Alzheimer's Association and another from Johns Hopkins, followed by a discussion with our moderator. Then there will be an opportunity for you, our audience, to ask questions to our presenters. After that, I'll return for a few announcements and bring the webinar to a close. So JoAnn, I will turn the webinar over to you momentarily. All right, there you are.
Okay, we're thrilled to have two very distinguished guest speakers for this evening, Doctors Heather Snyder and Dr. Abhay Moghekar, who will make short presentations, after which we will have a moderated discussion and an audience question and answer period with the experts. Our first speaker is Dr. Heather Snyder, Senior Vice President of Medical and Scientific Operations for the Alzheimer's Association. Let me give you a moment to read the slide about her role. Clearly, her advocacy for critical is important for critical to improving the quality of life for people at risk for developing and those living with dementia, with medical breakthroughs. So look at Dr. Heather's slide. Our second speaker is Dr. Abhay Moghekar, who will tell us about Alzheimer's biomarker research, which leads to diagnostic blood test development in the lab and how those discoveries move from bench to bedside. When Dr. Moghekar has finished his presentation, we will move into the discussion. Now it is my pleasure to introduce Dr. Heather Snyder.
Thank you so much, JoAnn, and thank you so much to all of you for being here with us tonight and for spending a little bit of time with us on this discussion as we talk about biomarkers, blood tests, and breakthroughs. And there's just so much happening in this space, and I know all of you are reading about it and seeing it in the many different places and many different media and headlines. And how do we break that down? How do we talk about that in a way as we think about what does it actually mean? And so we've seen so many stories around blood tests. Well, what does that really mean? And what it really comes down to is these are in many ways more accessible, more affordable tools that allow us to do accurate diagnosis. How do we get to making it more accessible to more individuals around the world? So just a few disclosures. I am a full-time employee of the Alzheimer's Association, and my husband works at Avid Labs in an unrelated field. So for those of you that don't know about who the Alzheimer's Association is, we are a science-based patient advocacy organization and a vision of a world without Alzheimer's disease and all other dementia. So while we are called the Alzheimer's Association, we actually represent all of the diseases that cause cognitive impairment and dementia. Now, I'm going to just really talk give some high-level definitions. Some of you may know all of this, but setting the stage for the rest of us as we have this conversation going forward. And we often hear these terms used interchangeably: dementia, Alzheimer's, vascular dementia. And what is really the difference? So dementia is the umbrella term that really describes the loss of memory, thinking, and other abilities that interfere with daily life. Alzheimer's disease is a cause of dementia. And when we think about Alzheimer's disease very specifically, it's defined by certain brain changes. When we look inside the brains of individuals, we see very specific things. We call them plaques and tangles. Plaques are clumps of proteins between the brain cells. Tangles are clumps of proteins within the brain cells. Plaques are made up of beta amyloid and other things. Tangles are made up of tau. And so we'll often talk about beta amyloid and tau, and you'll hear more about that as we talk tonight as well. And as we think about Alzheimer's disease, we now understand that the underlying biology of Alzheimer's is actually changing up to 20 years before someone has the memory changes that we have much more frequently associated with Alzheimer's. Of course, we have individuals that have mild, moderate, and severe Alzheimer's. So that's dementia due to Alzheimer's disease. And there's also a window of time where individuals may experience cognitive decline due to Alzheimer's, mild cognitive impairment, but they're still able to do many of the things within their daily life. And so this idea of Alzheimer's disease as a continuum has opened up the window for us to understand, well, what are those earliest biological changes and how do we measure them? So if you want to think about like diabetes, we think about diabetes, well, you often might see a prequel to changes of diabetes with your H1C, or heart disease, cholesterol. That's something that's measured really throughout our entire lives. Well, elevated cholesterol could be a sign of underlying biology and on that continuum of what we call heart disease. So those are similar ideas. When we think about the Alzheimer's disease continuum, what are those earliest changes and how can we measure them? Because we want to be able to intervene, whether with medication, behavioral modifications, combination approaches at that earliest time point, and stop, slow, or prevent or delay that continuum, the decline in loss of cognition. There are a number of medications that are approved for Alzheimer's disease, and I'm going to walk us through this slide because it's a little bit busy. So if you look at those that are here in the orange, these are medications that treat the symptoms. So there's no evidence that they are impacting the underlying biology of the disease as the disease progresses. These are primarily approved for individuals that have mild dementia, moderate dementia, or the more advanced stages. Those here that are in the like cyan color that you see, or the aqua color, these are mostly medications that target mood or behaviors. We know many individuals with dementia experience what we call non-cognitive symptoms. So changes maybe in sleep or agitation or apathy and those types of things. And these medications are really targeting the behavior to improve overall quality of life for the individual. And those here in purple, these are the newest class of medications. These are targeting the underlying biology. So I will note that the one from 2021 is no longer available, but we have two new medications in the last 2 years that have been approved in the United States by the Food and Drug Administration. And these target the underlying biology, lecanemab and donanemab that you see here. So a little bit about these, because I know there's often a lot of questions. And this has also changed the conversation of early detection, although the Alzheimer's Association has been a staunch supporter that early detection has always been incredibly important. And this has added a new reason as to why early detection is important. Lecanemab and donanemab were tested in individuals with mild cognitive impairment due to Alzheimer's or mild dementia due to Alzheimer's. So these are the earlier stages of the disease. And it requires that there is confirmation of the underlying biology in an individual. So that requires the use of a biomarker or a measure of that underlying biology as well. And primarily it's been linked to the beta amyloid plaques. That is one of those hallmark brain changes that I had mentioned. Now, I did just want to give a little bio 101 for all of us on the phone. We talk a lot about beta amyloid, or you can see it depicted here as A beta. And we talk a lot about it as one single species, but actually it exists in an incredibly dynamic state. Sometimes it's sitting alone, sometimes it's in a small group, sometimes it's in a much larger group, like what we would call a plaque. Sometimes it might change its shape, it might stand or it might sit, if you will. So it looks like these little spaghetti noodles or lasagna noodles. And all of these are representative of different states that beta amyloid might exist in. And when we think about these two medications, they both are targeting, the ones that are targeting the underlying biology, they're targeting beta amyloid. And we talk about that as the same thing, but in fact, they're a little bit different. So lecanemab, one of the medications that's been approved targeting the underlying biology, it's primarily developed to target this form of beta amyloid, although it has also been shown to bind to other forms and help remove them, lowering the levels from the brain. The other medication that was approved is called donanemab. And this was developed to really target and go after the beta amyloid that's present in these plaques, in these large clumps of protein that we see in the brain, and again, help to remove them out of the brain and lower their levels. And both of these medications showed a benefit in terms of the clinical outcomes of individuals, both cognition and function, for those that received medication versus those that did not. But just important to understand what these are actually targeting and where they are. So as we think about these two medications, these are the two that have just been approved, and that's really important. And they're targeting that earlier stage of the disease, which is why an accurate and early diagnosis is so incredibly important. But I do want to underscore to this group that this is just part of the story. And as we understand more and more about the underlying biology, we've seen an incredible growth in what's moving into clinical trials and really targeting the diverse biology that's happening. Alzheimer's disease is not a one single path or one single biology. And in fact, it's an incredibly complicated disease in a complicated organ, our brain. We know there are changes in our immune system, our vascular system, the bioenergetics, how our cells are making and producing energy. Brain cells are talking to one another. And all of these are mechanisms or biology that we see changed in Alzheimer's, and all represent an opportunity for us to think about potential treatments or potentially measuring as a biomarker, as a measure of that biology. And in fact, that's what we're seeing moving into clinical trials. Based on an annual report, there's over 127 unique therapies in 164 clinical trials. And I think what's incredibly exciting is when you look at this, 75% of these are targeting novel disease processes beyond the amyloid and beyond the tau. And that's really the evolution of our space in the field. And as many of these, as we think about that full spectrum and that continuum of Alzheimer's that we talked about, it's really important that we have early and accurate diagnosis so that we're able to really think about that personalized approach. What's your biology? How do we best treat you based on that biology? And so, for a long time, there have been these benefits of an early and accurate diagnosis. These are not new, but they are underscored with now this potential access to treatment going forward. So again, if there's the medical benefit of the access to current treatments, there's an opportunity to participate in a clinical trial, have that conversation, find that trial that might be right for you and participate in that. Thinking about what you might prioritize in your overall health. How do you want to manage your quality of life? Perhaps there's making lifestyle behavioral changes. We know, for instance, that individuals, even once they're experiencing cognitive changes that engage in greater levels of physical activity, seem to have better quality of life. And there's been a number of studies that have identified that. But we also look at the social and emotional benefits of an early and accurate diagnosis, understanding what's happening and why. Having that time to care for your own care future or your financial future with your family, and make those decisions for yourself. And it gives you that opportunity to access resources and put those in place for you or for your family, as well as planning those end of life decisions for yourself. And of course, there's a lot of evidence to suggest that with an early and accurate diagnosis, particularly individuals that may have other medical conditions, this allows you to manage those conditions and has financial benefits to the families, especially, for instance, somebody has diabetes, they may forget to take their insulin. And you can manage that when you understand what's happening with their Alzheimer's disease. You can manage how to ensure that they're taking their medication and that is handled accordingly going forward. So when we think about how Alzheimer's disease is currently diagnosed, it's really important to understand that there's no single test that can determine if a person is living with Alzheimer's disease or another cause of the cognitive impairment or dementia. And clinicians today use a combination of tools that allow them to make this accurate diagnosis. Now, we know when we look at a physical exam and a neurological exam, just those two things alone, clinicians do relatively okay. About 70% were accurate in terms of the diagnosis. But adding in a measure that allows us to confirm the accuracy of what's happening and really give the additional confidence allows us to go to over 90%. And that's with these tools such as biomarkers. So what's a biomarker? A biomarker is measuring a biological change. They are reliable predictors of the underlying disease. So some examples, cholesterol is a biomarker for heart disease. Glucose is a biomarker for insulin resistance and diabetes. And these can be used as diagnostics. These can be used predictively, although we're not there yet in Alzheimer's. They can be used in terms of enrollment in a clinical trial as well, or understanding if someone's responding to a treatment. So there's lots of different ways that a biomarker can be used. And I know today we're primarily talking about them from the diagnostic perspective. And when we're looking at the space of Alzheimer's research, there's so much happening. And this has really expanded and exploded in the last 10 or 15 years. We have tools that allow us to, for instance, take a picture of the brain through imaging. There's two kinds of imaging. There's both PET imaging, which uses a tracer that goes in and lights up the brain in certain ways. There's also MRI, which can actually allow you to look at specific regions of the brain and rule out what may be causing some of the memory impairment for an individual as well. But more, I think what we've heard more about in the field, and I know many of you have heard more about, is this transition to fluid biomarkers. Because imaging is not as readily accessible, it can be very expensive and invasive for an individual, as can cerebral spinal fluid. Cerebral spinal fluid is the fluid that our brain is bathed in, and a lumbar puncture or a spinal tap is how you're able to measure that. But there are also so many things happening in the idea of a blood test and moving those things forward. The idea that we can look in our blood, a simple blood test will allow us to actually have a view into what's happening in the brain. But we're by no means done. There's actually a lot of other work that's happening. There's work happening in terms of skin biopsies or saliva that may be also helpful in future diagnosis. Retinal imaging, can our eyes be another window into what's happening in our brain? So all of these are spaces that are moving forward. And I know today we're going to really focus in much more specifically as we think about blood tests and where blood tests are, because we want to get to that point of what's the right test for the right patient at the right time. How can we make sure that we're diagnosing accurately at that earliest time point and giving an individual, the patient, the opportunity to make decisions about their care, accessing and addressing their care needs, including treatments such as those that I mentioned here tonight. And this really puts us into a landscape as we think about Alzheimer's disease. I mentioned there's so many things that are happening in the underlying biology. We have tools that allow us, such as imaging, taking a picture of the brain or looking in plasma, which is part of our blood, to look at, can we measure those changes in an individual? Can we then personalize our treatment, our intervention, whether it be with medication or whether it be with other behavioral modifications and thinking about the combination approaches, because it's only going to be through this combination that will have sustainable strategies and effective treatments for all individuals going forward. So I did want to end before I pass it to my co-presenter today with some resources that are available, including a lot of information in where to get access to information and other resources in the community. But I did want to note information about ongoing research and where things are can be available on the Alzheimer's Association's website, as well as a tool to help you identify what are the clinical trials in your community and identifying what might be the right trial for you, or perhaps you live in Maryland or Virginia, but your family member lives in Chicago or California. You can identify clinical trials that are right for them in that place as well. And so with that, I really appreciate all of the time and you listening to me for a few minutes. And I'm thrilled to present, to pass it to my co-presenter today, Abhay Moghekar, who's going to, from Johns Hopkins School of Medicine, who is going to present a little bit more detail in terms of the blood-based biomarker and some of the exciting work that's happening at Johns Hopkins. Abhay?
Thank you, Dr. Snyder. That was an excellent overview on the importance, the need of these biomarkers, as well as the exciting work being done in the Alzheimer's disease area, not just focused on these few initial targets. So it's a very exciting time to be taking care of patients, because for a long time we did not have any concrete help to offer to these patients, and now things look promising. And one of the things that's making it possible are these biomarkers, and I'm going to specifically talk about some biomarkers in blood. So I'm going to talk about many specific products, but I want to be clear, I'm not endorsing any one of them in particular. I'm just providing this for educational information only. I mean, I am going to be talking a bit of some blood and some CSF biomarkers, and Fujiri Bio is one of the manufacturers of these. They provide research support to my lab. So what are the established AD biomarkers that are already FDA approved now? So there are some in cerebrospinal fluid that target amyloid beta, tau, and a phosphorylated form of tau. And these are obtained by a spinal tap. And then there are PET scans that you can do that target amyloid and tau also. So what is cerebrospinal fluid? So cerebrospinal fluid is a fluid that bathes the brain, surrounds the spinal cord, and extends all the way down into your spine. And it can be sampled from the base of your spine, right here, where there is no spinal cord. So it's relatively easy to sample it without causing any injury. And you can measure levels of amyloid and tau in the protein in the fluid, and those are reflective of what's happening in the brain. Similarly, PET scans are available now. There are 3 FDA-approved ligands for amyloid, and there's 1 FDA-approved ligand for tau. And as you can see, the amount of tau deposition in a patient with Alzheimer's disease is high compared to a normal control. So why are biomarkers important? So one, Heather has already gone over this, but again, to emphasize that even in advanced specialty tertiary clinics, we get the diagnosis wrong if we just base the diagnosis on a clinical assessment around 25 to 30% of the time. And this rate of misdiagnosis is likely even higher in primary care settings. This diagnosis is also more challenging in the earliest phase of the disease when patients have only mild cognitive symptoms. So all these challenges existed until biomarkers came along, and it made it more accurate to diagnose patients with Alzheimer's disease. These biomarkers are also important for prognosis, for predicting who will progress from MCI to dementia, at least in terms of CSF and PET. We're not quite there at blood yet. But these biomarkers are important for selection of patients for clinical trials, as was already mentioned. Many of the earlier trials in Alzheimer's disease failed because the correct patients with Alzheimer's disease were not chosen because the diagnosis was based on a clinical assessment and not on the actual biomarker measurements. These biomarkers can also be used to track, is the treatment having its intended effect? And I'll go over some examples later. So we already have established FDA-approved biomarkers in the clinic right now, but there are several problems with these markers. One, you need either specialized PET centers. There are only a handful of them around the country that can do this. PET involves exposure to some amount of radiation. And it's really not scalable at a population level. Millions of people are affected with Alzheimer's disease, and we don't have as many PET scanners to do this. And even though these are FDA approved, they are still very expensive. And despite insurance now covering them, there's a 20% copay. So the out-of-pocket costs are quite high still. In contrast, CSF, you need specialized skills. You need a neurologist or a trained neuroradiologist to do this. It cannot be done in all individuals, especially patients who are on blood thinners. And again, it's not scalable. There are not enough neurologists in the country to do all the patients with Alzheimer's disease. And while they're less expensive than PET, they're still more expensive than your routine blood test that you do. So we need blood biomarkers because we need so that any trained personnel, a regular phlebotomist, your local pathology lab can be able to do this easily and send it off to a lab for testing. Blood testing will be less expensive than CSF or PET, and blood testing can be scaled to the entire population and potentially in the future be used even as a screening tool. So development of blood biomarkers is very important and needed. So why haven't we done that yet? So a little bit of an analogy. So you should think of biomarkers as tiny messengers floating in your blood. And these messengers are carrying messages from the brain. But these messengers are present at very low concentrations in plasma. So plasma has thousands of different proteins, the most abundant one being albumin. So if you look at the concentration of albumin, it is several 100,000-fold higher than that of A beta 42, PETA, or NFL. So it's equivalent to trying to detect 100 grains of sand in 17.3 metric tons of sand. So that is a huge challenge. And that has implications for the quality of these tests too. These tests better be very accurate such that fluctuations in these messages should not affect your diagnosis at the end. But thankfully, there have been development of newer technologies that enable us to detect this, minuscule amounts of these protein amidst millions of protein molecules floating in blood now quite accurately. So blood biomarkers have focused initially on amyloid beta, which forms plaques, but also followed by phosphorylated tau, which is a component of the tau tangles. And there are other biomarkers like NFL and GFAP that look at different aspects of AD pathology. Now, if you go to your primary care physician or a tertiary care center, if you have cognitive impairment, and your doctor can order these tests now through these different labs. But we have to be very careful of interpreting how these different labs come out with the thresholds, meaning how do they define who is normal and abnormal? None of these tests are FDA approved. So even though all of them are available, they're not quite FDA approved for blood biomarkers. But we expect them to be approved soon. What is the problem with plasma amyloid markers? Unlike in CSF, where patients with Alzheimer's disease have a 40 to 60% difference in their values between normal and Alzheimer's disease. In blood, the difference is, only 10 to 15%. And 10 to 15% sometimes is just a day-to-day variation and lab testing. So it's a very small signal amidst a lot of noise. In addition, amyloid is produced by peripheral tissues, the other organs in your body that make this amyloid. So the brain signal gets diluted. So currently, it's not regarded as a robust test for brain amyloid, unlike other tests. By other tests, we come to phosphorylated tau. So unlike the very small differences seen in amyloid beta 42 and 40 between controls and Alzheimer's disease, phosphorylated tau, and there are different forms of it, 181, 217, 231, they are increased 2.5 to 6 times that in Alzheimer's disease as compared to controls and other dementia. So the signal is very, very clear. And in multiple studies of all of these different isoforms that were tested, p-tau 217 seems to be very promising. And in fact, in many studies have been found to be comparable to CSF and tau PET. But it's not quite intuitive. Many researchers now strongly believe that these plasma p-tau assays mostly reflect amyloid aggregates, and I won't get into the biology of why that is, but suffice to know that phosphorylated tau isoforms in plasma and CSF are a better reflection of amyloid tangles in your brain rather than tau tangles. There are different phosphorylated tau molecules like p-tau 205 and MTBR 243 that in fact do correlate well with tau tangles in the brain. But those again are not ready for prime time yet. It's very important not to forget the context of use. So far, all the research has focused on assessing these blood biomarkers in patients who have cognitive impairment as ascertained in secondary and tertiary care centers. The utility of these markers in patients without symptoms or without any objective confirmation of cognitive deficits is not known. It's clearly an area of active research, but we are not quite there yet. Currently, these blood biomarkers have been tested and validated only in secondary and tertiary care centers and only in patients who currently have cognitive impairment. There's active research going on in testing these in primary care centers across the world right now. But you have to be, one other thing to be cautious about is, unlike CSF and PET test results, which are a clear yes or no, blood biomarker tests are all going to yield tiered results. And this is where it becomes important that your doctor who is ordering these tests is well versed in interpreting how these blood biomarker test results come out. Meaning, if you have cognitive impairment, you do this blood test, there are going to be two cutoffs, not one. So it's not going to be a yes or no. It's going to be a yes, maybe, and no. And by that, I mean, if it's a positive test, if it's clearly above the high cutoff, you're going to be positive for the test, which means it confirms you have amyloid pathology in your brain. If it is below the lower cutoff, that means they've ruled out Alzheimer's disease. But if you're in this intermediate range, and that an ideal test should result in only 15 to 20% people falling in this intermediate range, that means the blood biomarker test is not conclusive, and you need to get additional testing to either confirm whether you have it or you don't have it. So this nuance of interpreting blood biomarker tests is very important, and you need a trained physician. So your general practitioner who may not have as much knowledge about Alzheimer's disease and is not well versed in interpreting these blood tests may not be the right person to go to get these tests done. In addition, many of these tests may result in uncertainty, and the number of subjects with uncertainty will be higher in patients who just have subjective cognitive symptoms, meaning they don't have objective impairment. We expect many of them to be intermediate and will need additional testing with either PET or CSF, which are best done in tertiary care centers. And many of these patients with underlying intermediate probabilities, even PET and CSF may sometimes not be conclusive. And then you have to wait and follow up, and you may need repeat testing in 12 to 16 months. So I just want to be cautious that, this is going to be a very cautious rollout and a nuanced interpretation when these tests come to market. And one of the reasons for caution is that we still don't fully understand how these blood biomarker levels are influenced by how old you are, whether you have kidney disease, diabetes, coronary artery disease, your body habitus, your ethnicity. Some preliminary studies show that all these influence the level of blood biomarkers. And if that's the case, do they change the cutoffs when somebody is called low probability or high probability? And again, this is an area of active research. But in addition to those factors, there are pre-analytic factors. So it's essentially, did you fast when you have the blood drawn? What kind of tube was it drawn in? How long before it reached the lab? Was it frozen correctly? Was it filled correctly? What was the centrifugation speed and temperature? All these parameters can affect these blood biomarkers. And these are all being studied actively to know which of them significantly would change your diagnosis, say from high to low or low to intermediate and so on. So further research is still needed. We need to obtain what are the thresholds in diverse populations, people of different race and ethnicity. We need to understand how these other comorbid conditions, kidney disease, diabetes, heart disease, affect the cutoffs for these biomarkers. Is there a difference depending on the time of day you draw it, or what is the day-to-day variation? That's still being researched. I mentioned the pre-analytical variation, and we need cutoffs that are widely applicable. If you have different cutoffs for different populations, it becomes challenging to interpret them and implement them in a primary care clinic. So these prospective studies are, in fact, going on right now all over the world. But in the not too distant future, I envision eventually these will get approved in primary care. And the goal then is just like you go and to your doctor in your 40s and 50s and get your cholesterol tested. And if it's high, you get started on a statin. I think blood biomarkers will eventually reach that stage where if you have cognitive symptoms, you'll get the blood test, you'll get a probability of high, medium, or low. If you have high probability, you may jump directly to a disease-modifying treatment without needing CSF or PET studies. Again, we are not there yet. This is a future. And if you're low, they've excluded it, and your primary care physician can reassure you, no need to worry, we'll see you back again. And these biomarkers, as Dr. Snyder mentioned, are also going to be very useful in clinical trials because they can help us select the right population, meaning if there's an at-risk population of with cognitive impairment, these blood biomarkers can select patients who have Alzheimer's disease. They can enrich that even further. Say you wanted people with only high tau, so these blood biomarkers can be used eventually to select people with high tau or low tau, for example. They can also be used to monitor the effect of the treatment by measuring the effect on the change in biomarkers. Say you have an antibody to tau, you could monitor its effect on blood tau levels, for example. So in addition to being useful in diagnosis, they play a very important role in clinical trials. And with that, I'm going to end my presentation. And some of these trials are in fact being done at the Alzheimer's Disease Research Center at Johns Hopkins. You're welcome to participate in them. It can be as easy as being part of an observational study, where no intervention is done, to testing out different lifestyle intervention changes and even medications if you actually have the disease. With that, I'm going to stop. Thank you.
Thank you both for those great presentations. The audience, please drop your questions in the chat, in the Q&A box while Corinne assesses those. The Brain Matters team have prepared a few questions for our speakers. So drop your questions in the Q&A box and we'll proceed with some prepared questions. And to Heather, Dr. Snyder, the first question, what is the comfort level of using blood tests and interpreting them among primary care doctors with patients in their offices? And what is needed and what are the next steps?
Yeah, such an important question, and such a question we get all the time at the Alzheimer's Association. At this point in time, as you heard, many of these are not at that point of translating into primary care. They are really only being used in specialty care as part of a larger workup, as part of what that looks like. There is an ongoing effort right now in terms of developing specific guidelines that help inform how these types of blood tests would be used in clinical care, starting with specialty care, because that's where they're being used now, and providing that sort of guidance. And then as the evidence builds of how these will translate, what these really mean when we're in larger and real world populations, and how they're performing, we'll see that translate into primary care and see that those guidelines evolve. So not quite there yet in terms of primary care, at least in most instances. We're mostly seeing them being used in specialty care.
Thank you for that answer. How do they cover these diagnostic tools, and will it be very costly to access them?
Yeah, another and really incredibly important question. And so when we think about the imaging tools, there's actually been about a decade of work that has now resulted in the Centers for Medicare and Medicaid Services now does cover the use of the imaging tools, the amyloid imaging and the tau PET, as well as FDG PET. And so there's a lot of work that's gone into making those more accessible, although PET imaging is still in and of itself not necessarily accessible to everybody for a number of reasons. And some of that, a big part of that is geography. But as we also said, that's just a much more expensive. There's a large copay. It's a much more expensive and more invasive process to go through. In terms of the blood tests, because as you heard, they are not FDA approved at this point in time. There is not a coverage path in most instances for these, but there is a lot of work to make sure that as we get that kind of that final data and really understand how these work in the real world, as that data and information come forward, there's a lot of effort at this point in time to ensure that then these are covered for the accessibility for all patients.
I'm going to Dr. Moghekar. What do the new diagnostic tools like blood-based biomarkers change about the accuracy of a patient's diagnosis and prognosis?
So that's a great question. So two things. I mean, as compared to CSF and PET scans, blood biomarkers are now equivalent to those. So if they're as accurate as CSF and PET, and they are more widely available and less invasive, and hopefully will be covered by insurance soon, we see there being a wide rollout and this being available to everyone, all over the US and not just at specialty care centers. So it will essentially democratize the diagnosis of Alzheimer's disease and make it available to the wider population.
Thank you. And how are blood-based biomarkers connected to improving early dementia detection?
Again, many patients who are seen in primary care clinics and they go with mild cognitive symptoms, right now they don't have a clear pathway to get a firm underlying diagnosis of Alzheimer's disease. And it takes time to refer them to a tertiary care center. As everyone knows, there's a long way to get into these tertiary care centers. So that can actually delay the diagnosis of Alzheimer's disease. So eventually, hopefully, when we have these blood tests, the primary care physician can himself or herself directly order these tests and be more assured of the diagnosis or rule it out. So I think we will get to early detection because of these blood tests.
Okay, thank you. Thank you. And these next questions could be for either of you to answer. Do people from different backgrounds, such as social determinants of health and disparities like race, ethnicity, gender, socioeconomic status, sexual orientation, have different biomarkers?
Again, very important question. And I know this is an area of quite intensive research at this point, because most of these studies that have been conducted in blood biomarkers were in Caucasians, and we definitely need to validate them in the people with different socioeconomic strata, people with different racial and ethnic backgrounds, to make sure that the accuracy is not compromised in these populations. There was a study that looked at patients with all-cause dementia in different racial backgrounds, and the level of AD biomarkers in patients with diverse ethnicities was different than that in Caucasians, which would suggest that they may have a higher burden of other conditions like vascular dementia, for example. So is the performance of these blood biomarkers going to be the same across all these different backgrounds? Is important, needs to be done, and is being done.
Yeah, and maybe just to add a little bit to that is there's some in some of the ongoing large phase 3 trials, there's been an opportunity to really leverage both ensuring broader community engagement, and so an increase in representation in the screening of potential participants for these studies. And what this has also offered is an opportunity for us to better understand how these blood-based biomarkers work in inclusive populations more broadly. And I think just to kind of underscore that part, it's maybe likely that what we thought of as or referred to as Alzheimer's disease is maybe not what is causing the cognitive impairment that individuals are experiencing. We don't know. And I think that's part of what we really need to understand. And that's where that translation to real world care needs to happen as we think about these blood tests. But it also underscores the need for us to continue to investigate and research what is causing the cognitive impairment. Because when we think about that personal approach, we want to know what's your biology, what's changing, and what can we target as a potential therapy. And so, or intervention, and maybe that's behavioral or lifestyle modification. Maybe that is medication. Maybe that's combinations like we think about for heart disease. The only other thing I would note is the biomarkers are a representation of the biology, and there's no race is not a biological construct. And so we separate those things out in that way. But it's really important that we understand what is happening in all individuals, that we can measure it, and that we can help treat them for whatever might be causing the cognitive impairment.
Okay, thank you both. It seems like we have some questions from the audience. So Corinne, would you take over here and look at the questions from the audience?
Absolutely. Thank you so much. And thank you to both Abhay and Heather for their great presentations. I'm going to just try to loop in a couple of audience questions because there are some really excellent ones out there. I want to start with one that gets at different types of dementia. So Dr. Snyder and Dr. Moghekar, you both touched on this in some way. And several of the questions are asking about how these different markers might help understand the type of dementia someone has and whether they can differentiate whether someone has, for example, Alzheimer's versus frontotemporal versus vascular disease as the cause of their dementia. And then as a secondary question, a related question, whether blood biomarkers can be better indicators of dementia risk than genetics, maybe ApoE genetic status. So two related questions. I don't know who would like to go first.
Yeah, no, I'm happy to start, and let's see if I can remember both questions. So, I think maybe start with the second one. So, is our biomarkers better or inform more than maybe ApoE status? So it's important to understand ApoE status, or ApoE, which is, for those of you on the call, it's a gene that has been associated with increased risk. So we all get one copy from each of our parents of our genes. If you have one copy, you have an elevated risk, two copies, an even greater risk. But it is a risk gene. So there are people that are living with two copies of ApoE4 that do not have Alzheimer's. There are individuals living with Alzheimer's who do not have ApoE4. So ApoE4, or having one or two copies of ApoE4, is in no way of being definitively predictive of whether someone is going to get Alzheimer's or not. And so it adds to your risk, and that's important, but there are lots of things that may be part of that risk. And in fact, when we look at our genes, there's been over 100 different genes and variations of genes that have been associated with both increasing and decreasing a person's risk. Biomarkers are a little bit different in that is representing the underlying biology that is present in an individual. The information today is there is no group that recommends that biomarkers should be tested in individuals that are not experiencing cognitive symptoms. And biomarkers in terms of clinical care should be used in the context of helping to inform an accurate diagnosis and really helping to make decisions around care planning, including treatment going forward. And so that's that part. And the second question, and then Abhay, you can add in either or both, is the second question is, this is part of why this is such an exciting time. And I hope we've done a good job of sharing that with you, as we've talked very specifically here about Alzheimer's biomarkers, but there is some amazing work happening across all of the diseases that cause cognitive impairment. There's a lot of work happening with, for instance, a tool that allows you to measure another brain change that's associated with Parkinson's disease and Lewy body dementia. And so that work has really suggested and given us a huge amount of better understanding of what might be some of the brain changes present in individuals, and also told us in many individuals with Alzheimer's, they may also have Lewy body changes. So when we think about what's your biology and how do we personalize and treat you, these are going to be really important tools. The same is true. There's some new work that's happening in the FTD space, the frontal temporal dementia space. There's some other work that I know the Alzheimer's Association is funding at Mayo Clinic that's looking at dementia with Lewy body biomarkers. And so this whole space. the biomarkers with Alzheimer's are probably the furthest along, but that by no means does not, there's all of these spaces are going forward.
I mean, the only thing I would add to that is autopsy studies now tell us that, in fact, pure Alzheimer's disease is not that common. Often people have a bit of amyloid, a bit of tau, a bit of vascular injury, microvascular disease. They have a little bit of Lewy body pathology. In fact, 30% of patients with Alzheimer's disease develop Lewy body pathology. 30% of patients with Alzheimer's disease have TDP-43 pathology. So as we get older and older, we accumulate all these mixed pathologies, as I would like to call. And in that case, if you targeted just one pathology, you shouldn't be surprised if a patient may not respond, because there are other pathologies that you're not actually targeting. And as Heather said, there are biomarkers being developed for all these 4 different proteins in CSF first, definitely moving on to blood for more wider application. In younger people, pure pathologies are likely, but as we get into the 70s and 80s, mixed pathologies become much more likely. And that's why we need biomarkers for all of those pathologies if we hope to treat dementia at a personalized level.
Thank you both. That's a great response, very comprehensive. And you both remember both my questions, so that was perfect. Dr. Moghekar, I want to build on something you just spoke about. So we just heard that we don't yet have biomarkers for these other types of dementia, but you also talked about we know that individuals have different types of pathology when they have dementia. Can you touch a little bit on how we know that if we don't yet have biomarkers for it?
I mean, one of the ways that has been possible to figure out that patients have these multiple pathologies is because these people have very generously donated their brain, so that when they pass away, they donate their brain, and we can then do a histopathological analysis, where we do tiny slices of the brain, and we stain these slices of brain with antibodies that are very specific to these different misfolded proteins in the brain. And there are these ADRCs, Alzheimer's Disease Research Centers, all over the country now, where anybody can enroll and be followed longitudinally with cognitive testing, blood biomarkers, imaging studies. And eventually, if you contribute your brain for research, we develop a much deeper understanding of all the different pathologies that contribute to dementia. So that's how we know that people are having all these different pathologies, is because of their kindness and generosity.
Thank you for clarifying. I think given the time, we're at 7:55, and I know we want to stay on time. We'll stop answering. There's a couple of questions in the chat. Maybe if you could both look in there, and ones we didn't talk about, that would be great. I answered a couple. Thank you for taking the time to answer our audience questions and for your great presentations. And I'm going to pass the floor back over to Tony so we can move to announcements and bring the webinar to a close.
Okay, I'm hoping you can hear me. It looks like I'm off mute, so we're good. In about 2 weeks, you will receive a recording of this webinar. I noticed someone asked about getting the slides. The slides are going to be part of the webinar recording that you receive, and you'll receive a takeaway sheet with some main points to it. Our next webinar will probably be in next spring or summer. A couple of quick announcements. The new Brain Matters stakeholder we're very proud to announce this, is the Caucus of African American Leaders of Anne Arundel County. We're so delighted to have them on board and thank them for their presence and co-sponsorship of these webinars. Also, there's going to be a forthcoming Global Council on Brain Health report, probably this summer, that's going to be about exactly this topic. So we'll send you the Global Council on Brain Health's website so you can track what's going on and take a look at some of their other reports on brain health and related matters. Additionally, if you're looking for a great holiday gift for a youth, then consider this. I Can Dream Too: How to Talk to Children About Dementia is actually written, co-authored by one of our Brain Matters stakeholders with Delta Sigma Theta in Columbia. Here's a picture of the cover of the book. It's really a great book if someone is young, doesn't understand what's going on with a loved one, like a grandparent or a great uncle or something like that, who shows up at a family event over the holidays and seems different. So this is a good way to introduce children to what's happening in their loved one's life. I'd also like to take a moment to acknowledge and thank everyone on the Brain Matters Steering Committee who makes this webinar possible. We spend months in advance planning and sorting through things right up until the last minute and making sure it's a good webinar for you. So I wanted to say a special thanks to Dr. Snyder and Moghekar for sharing your knowledge and information and resources with us. And most importantly, I want to thank you, the audience, for joining us. You're why we do this. Your brain matters. Consider being part of one of these newly emerging clinical studies or trials. Contact us if you have any questions about that, or take a look at the Alzheimer's Association's Trial Finder. It's a good way to get involved, and maybe you can be part of the next breakthrough. So thanks again, everyone. It was delightful to have you here this evening. I hope you learned something, and I wish you a very safe and happy holiday season. Take care.